everybody. It's so nice to see all of these faces today. To everybody, welcome back to our PMSF We Are Family Conference Scientific Sessions. We have a fantastic session today focused on meeting the Scientific Advisory Committee. As you may know, we have this advisory committee that consists of MDs, PhDs, and MD PhDs from around the world. And we are so happy to be working with them and get updates from them pretty frequently. So before we get started, I'm going to share a brief word from our sponsor. So just 1 second as I pull that up, and then I will introduce the session.
Our mission at Jaguar is to accelerate breakthroughs in gene therapy for patients with severe genetic diseases.
With gene therapy, we can give the opportunity at a better life. That's what makes Jaguar special.
All right, so to get started today and introduce the session, some of you may not know that the Scientific Advisory Committee actually meets quarterly, and we share updates and brainstorming for the future. And we really wanted to invite you all to see this behind the scenes. We get to see it frequently, and it's really inspiring. So we're happy that families could join this session. And for families that could not join in person or at this time, we will have recordings available. So many people will benefit from this after the fact. So this group focuses on a range of research, all the way from projects, using cell and animal models to find out new information about the disorder, all the way up to clinical trials and very translational work. So we have quite a range of work. Please also note that the Scientific Advisory Committee is separate from the Medical Advisory Committee, with the exception of Billy Bennett. So the Medical Advisory Committee is known for advising on specific medical issues and sees a lot of patients, although we certainly have people on our Scientific Advisory Committee that also see patients. And the Scientific Advisory Committee is intimately involved also with a lot of our projects. So a lot of the things that we do, we run by them. So if we have a clinical trial that's upcoming, we might circulate information about that company or about the trial to this group before circulating for recruitment to you all to make sure that your best interests are at heart. So that's just one example, but we're in pretty frequent contact with the Scientific Advisory Committee. So the way this session is going to work is I'm going to share one very brief slide of updates from PMSF to the Scientific Advisory Committee, and then we will go around and have each person introduce themselves really briefly, where in the world you are, what connection your work has to PMS research, and specifically, what's so important about it for the families on the call. And we will start with a couple of people who have told me they have to leave early. So if there are more people that might have to leave early, please message me in the chat and we will coordinate that. And so we will go around right after I share a couple of updates. And then if there is time, we will take questions. We know that this is a very busy session with a lot of people, and we're trying to stay on time, especially with different time zones. So we may not get to questions, but please put them in the Q&A anyway, and we will make sure they are logged and addressed. So thank you all for being here. It's so great to see all of your faces. 1 second while I pull up the updates from PMSF. Can you all see this? Okay, great. So these are just some recent updates over the past couple of months. I'm going to go through them quickly because I really want to give you all the floor. I also want to mention that every single piece of information here will be circulated in an email to the Scientific Advisory Committee afterwards, with attachments to some of major announcements in case you would like to circulate this to your communities. So the first one is, as you all know, we've been working on a specific ICD-10 application for Phelan-McDermid syndrome. We are happy to be in the very late stages of this process. For those who may not know or want more information, an ICD-10 code is essentially a label in the medical system for tracking the disorder and data associated with the syndrome. So right now, unfortunately, Phelan-McDermid syndrome is attached, grouped with about 50 plus other syndromes and disorders. That is a very vague category, so it's hard to track information like public health data, information for insurance and other services that may be covered, and also for pharmaceutical companies to really, find our disorder and take it seriously. So we hired a coding expert. We all submitted an application with the review of the Scientific Advisory Committee to the CDC. We were very lucky to get through that phase. A lot of rare disease groups do not. And we had Dr. Curtis Rogers present to the CDC in September. And now we are in a little bit of a waiting stage to hear back about this, but we are just thrilled to be moving forward and for all of your support on this process, including Curtis, who did a fantastic job. Second item is we just had an externally led patient-focused drug development meeting with the FDA last week. We were partners with CureShank on this, and it's essentially a way for families to communicate to the FDA some of their biggest concerns, biggest gaps, and their perspective, which will be compiled into a voice of the patient report to come, and that will be circulated to you all. And this helps the FDA make decisions about drug approvals, and it also helps pharma companies, in their design of clinical trials. So we're really thrilled. That was a beautiful day, and it was a lot of great feedback and stories from families, and that is also recorded and on YouTube. So we will circulate that as well in case you don't have that. Another item is that we've been in contact at PMSF with an Australia-based pharmaceutical company who just added PMS to their pipeline. This is PYC Therapeutics. They had a press release that we circulated a little while ago, but we will recirculate this as well. And this is thought to specifically target SHANK3 to increase it in an antisense oligonucleotide fashion. So you all will be receiving more information as we continue to meet with PYC about this. We have publications coming up from 2 PMSF conferences. Dr. Bennett or Billy might talk about this a little bit, but we had the Candid GI conference and also a genetics conference that are both being translated into meaningful publications and future work with the community on both of these things. Another thing that's really important for the Scientific Advisory Committee to be aware of is that we are starting a new grants program in 2023. We will likely open 2 new annual grants to fund research. We have a couple of categories for this. We're not announcing them just yet, but maybe in the next couple of weeks. And this will be open to you all to apply to. And we also are going to try to work out a schedule for reviewing and possible conflict of interest there with SAC and MAC members to judge these, but we will work on that. Another thing for some of the more clinical folks is that we've been hearing from a lot of families specifically that there is often difficulty getting physicians in urgent care or in hospitals to listen to really PMS specific recommendations about neuropsychiatric care, specifically with the use of antipsychotics. So the Medical Advisory Committee has written a information sheet that can be shared with providers. And we heard from families that the best way to share information like this is possibly through QR codes. So we will have these on our website and distributed to those, at least in the US, with cards that can be like little business cards with QR codes. So that is coming up. I'm sorry I'm taking so long, I'm hurrying up. So the US Clinical Consensus Guidelines is also wrapping up for publication. And I know there is a European counterpart to that, so we'd love to hear more about that in this meeting. And then this is one of our last science sessions for our year-long family conference at PMSF. So the recordings of all of the sessions in science, which covers about 12 different things of sleep, GI, neuropsych, clinical trials, will all be available as resources for you all to also circulate to your communities. And with that, I will stop sharing and stop taking up time. And are there any questions on any of that before we go around and do some updates?
Kate, did you mention how many people attended the PFDD?
It was the biggest engagement audience they have ever seen. I think it was over 400 people. A ton of families showed up, and other stakeholders. And so we've heard that it was one of the most successful engagement meetings they've had. They also, there was some polling that happened during it, and they actually had to adjust and not get any new answers because they actually tapped out the poll answers at one point. So not surprised because our community is so engaged. But any other questions or comments? Okay, so let's go ahead and get started. There were a couple of you who let me know you do have to run early. So I will start with you, and then if anybody else needs to run early, please private message me in the chat. But let's start with Dr. Audrey Thurm, who mentioned she might have to leave a little bit early today.
Thanks so much, Kate. I just want to start off by saying, oh my goodness, the foundation is doing so much. I mean, that is an incredible list of things that are all actually very recent or ongoing. So I just wanted to give kudos to you and to all the people, including a lot of the families that are making all these things happen.
Thank you so much.
So I'm going to try to share and not mess things up. How did I do? Did I do it? All right. So I'm just going to focus on a couple things. I think we're supposed to say like who we are and how we got involved in. Yeah. So I'm a clinical psychologist, and my focus is mostly on children and adolescents, but I've been in the field of autism and intellectual disabilities for a long time, and here at NIH. And one thing I want to make sure is clear that NIH is not just the people who give out the money. We also have a campus, and that's why this is on here in Bethesda, Maryland, where we do intramural research. And so I've been doing research on intellectual disability, autism, and genetic conditions for a while here. And starting in 2013, actually, I think we brought our first Phelan-McDermid patient that we started to do some natural history on our own, because again, at that time, there was no natural history. And then we joined forces with the larger effort that put in the network grant for the Rare Disease Clinical Research Network, which is part of NIH. So we do not get our money from them. We have our own NIH money. I know it's confusing, but we're part of the big effort to be a site here for the natural history study. And this has been going on now for since 2015. We're actually in the second round of it, but we're still recruiting new patients. So we want to make sure, I just want to spend a minute to explain that this study actually includes other conditions that it's being compared to, but this particular study is for mapping out the natural history of Phelan-McDermid syndrome. And I just want to say that even though we have a lot of literature now, we still, especially for adolescence and adulthood, have very, very little data out here on what's happening over time. And so this study is designed to look over time and to capture a large age group so that we can really understand how it unfolds. And I just want to emphasize here how exciting it is with clinical trials, and what you just mentioned, even more coming on the way, is that the FDA and the people working on these trials are relying on this data for the natural history. It's great that we've got anecdotes in the FDA and all those reports, but they're relying on studies that publish actually findings that Katie started doing a long time ago, but now are really looking over time at large groups. So I have information here about if you still want to participate, how you can get it, and an email, and we'll make sure to get that out. And the aims, at least with the first round, were really, it seems like so broad, but it's really just to characterize the study using longitudinal assessments. We did focus on biomarkers too, including MRI, and then really try to look deeply at the genetics in terms of how that relates to the phenotypes. So again, clarifying the phenotype, identifying the targets for treatment studies, right? Is that we all know there are many things we want to work on, but exactly what would be the best target based on the measures we can use is how the study can help. And then of course, a lot of it has been helpful towards the clinical practice and gone right towards the guidelines that are being developed. So this is just a few of the many, many publications that have related to the first round of the condition, including, the knowledge about the neuropsychiatric changes in adolescence that really we just saw very clearly, and move forward on with further studies from the 100 comprehensive in-person assessments that were done. And I will say that we also pivoted to telehealth in the pandemic, and I think there still is flexibility for how people can enroll and be engaged, and because we know that it's not easy, even with the travel funding that's provided to come sometimes. So telehealth is an option for some of it. In addition, at NIH, we took on a few topic areas, and we were able to do additional studies. And one was about the GI issues, which I won't speak to more than just saying that we did this. We also looked at overnight EEGs, which are not easy to do in research. And then another thing we observed that we're following up here at NIH on is the differential response to pain. And so, we are looking now, after seeing this in Phelan-McDermid and other conditions, at using imaging, which is just putting a cap on the head, not lying down anywhere. And so there can be movement, to be able to look at responses to pain stimuli. And we just very recently got IRB approval to start a separate protocol here that will be about looking at pain in individuals with all different genetic conditions with intellectual disability, because it's really important to include several to be looking at what differs and what's the same. And so patients with Phelan-McDermid are welcomed into this study in terms of looking at brain activation with pain stimuli. This is not recruiting yet, and it will only be local recruitment for this, but that's a large area. And I just wanted to make sure that was known. And I will stop because I think I'm past my time.
That was fantastic. Thank you. You were not past your time, just FYI, but that was perfect. And I know personally that Audrey is constantly working on new assessments that better represent Phelan-McDermid syndrome, and that there's just fantastic work going on with the natural history study and the larger group that focuses on that as well. As she mentioned, we do provide funding for travel to the study and other things related to the study. And all of the information about the natural history study is on our website. And I would love to connect more with Audrey later about some of the additional projects that seemed really interesting. So, and I should also mention that we're on a first name basis here. I apologize if that is, I'm just gonna, if that's okay with everyone, to just address everyone by their first names. So next up we have Alex. Alex said that he might have to run a little bit early, and I know that you had a fantastic publication that came out recently that I will be summarizing from families in our next quarterly research roundup, but take the floor, Alex.
Okay, thank you, Kate, for organizing this, and it's really nice to be here. And yeah, I'm an assistant professor in the Department of Neurobiology at the University of Utah. I've been my lab have been studying Phelan-McDermid syndrome since 2014 when it started. But before that, I was a postdoc at Stanford with Ricardo Dolmich, and that's when I started studying Phelan-McDermid syndrome using samples obtained from patients. And so what we do in my lab now, we have stem cells that were obtained from healthy control individuals and from patients with Phelan-McDermid syndrome. And huge thank you to all the families that participated and donated cells to our study. I think it was a few years ago, Joachim Hollmeyer and John Bernstein, they organized this collection, and many families participated. Yeah, so thank you very much for this. So we use the cells and we convert them into neurons because we don't have direct access to the neurons in the brain, and so we use induced pluripotent stem cells that are obtained actually from blood cells or from skin cells that were obtained originally from patients. So the steps is skin cells, stem cells, and then stem cell derived neurons. And then we can use neurons to look at cellular molecular deficits that are associated with the disease. We also, we found that excited resynaptic transmission is impaired in neurons, in stem cell derived neurons from patients as compared to healthy control individuals. Recently in my lab, we generated some engineered stem cell lines because, as you all know, Phelan-McDermid syndrome is heterogeneous in terms of genetic abnormality. Some patients, they have really small deletions or even mutations. Other patients, they have very large deletions. And in my lab, we generated this CRISPR-Cas engineered stem cell line where we deleted one complete copy of SHANK3. And we use this line together with control lines, and we studied cellular and molecular mechanisms associated with SHANK3 deletion using organoids and stem cell derived organoids. And organoids is a new approach to that people use to model disorders. What is unique about organoids is that they can produce different cell types that are present in the brain, not only neurons, but also astrocytes, some oligodendrocytes, different types of neural progenitors, both excitatory and inhibitory neurons. And they allow us to look into the dynamics of development. And we did recently experiments using these organoids, and we found some interesting deficits associated with SHANK3 deficiency. And my lab now following up on this study and trying to think how to intervene and rescue the deficits, and what we can do also to understand the relationship between the deficits we detected and synaptic deficits we also observed in this study and in the previous study. That's briefly what we do.
Thank you so much, Alex. As you mentioned, I think we all appreciate the extreme importance of these models that you're developing, and the better the model is for studying Phelan-McDermid syndrome, the better we can identify new targets and test new interventions. So we appreciate it, and the summary of your paper I will certainly have in the next research roundup, probably in about the next month or so. And this just, summarizes for families what the publication was about. Thank you so much. Is there anybody else who may have to run early that would like to go next? And otherwise, I might just do random. Nope. Okay. So I will just go with the first person on my screen and go that way. So that is Katie Phelan.
Okay, hi, I'm Katie Phelan. I am at Florida Cancer Specialists in Fort Myers, Florida. We had a hurricane about a month ago, but we've recovered, so we're safe. I am the director of the genetics lab here at Florida Cancer Specialists. So my section of the lab does mostly chromosome analysis and fluorescence in situ hybridization. We also have a next generation sequencing laboratory in the genetics department. So I've been involved with this syndrome since 1988, before it was a syndrome. We just had a case report, a newborn baby that we studied at the Greenwood Genetics Center who had a deletion of chromosome 22. And there really weren't any cases of simple deletions in the literature. So we reported it at a meeting that fall fall of '88. And then we wrote up a case report. And when I did the case report, I wanted somebody to do the molecular studies. And I found Heather McDermid, because I knew from the literature that she did molecular studies on a different syndrome, cat-eye syndrome. It's a duplication of 22. So that's how Heather McDermid and Katie Phelan got together. And rest is history. So no. So we did a case report that we published in the early 90s. And then families would contact me and by phone or by letter, because we didn't have the internet. And I started to learn about the syndrome because they wanted to talk about, or the deletion of 22q13 at the time. And they wanted to talk to the family that we had in Greenwood. So I would put the families in touch with each other. And the recurring theme was that the families wanted to meet other families because their doctors at home didn't know anything about the condition. And they were telling them all sorts of goofy things that even I knew weren't right, even though I wasn't a physician. And so after a few years, I applied for a grant to have a support group meeting in Greenville, South Carolina. And I roped Curtis Rogers into being our major clinical geneticist and his genetic counselor, Gail Stapleton. And we had a meeting at the Embassy Suites in Greenville. And the time I got the grant, I knew 9 families in the world who had this deletion. And by the time we had the meeting about 8 months later, we knew of 23 families, and 20 of the 23 families that we knew came to Greenville, South Carolina. We had a meeting. We had about 3 or 4 clinical geneticists who came and we did family histories and clinical genetic evaluations, medical exams on all these patients. We drew blood. For Heather McDermid was there. She took the blood back to characterize the deletions, and we started collecting clinical data on the families. So we decided to meet every 2 years, and we would do physical, genetic evaluations. So we started basically collecting longitudinal data on the kids. And at that meeting, I think the oldest, we had one individual in his 20s, we had a 15-year-old, and everybody else was under 10 years of age. So at a really low age range. And so we didn't know about a lot of things. We didn't know about seizures. We didn't know about kidney abnormalities. We certainly didn't know about regression. So over the years, that's kind of what I've been focused on, characterizing the cause of the syndrome, the deletions, the rearrangements. Heather McDermid and her group looked at the region of overlap. And once SHANK3 was described, that was in the interesting region of overlap. And then looking at the clinical features of the syndrome. So that's kind of where I've spent a lot of my time. What else have I done? And then I like family support. Of course, that's always been a great interest of mine. So having the support group meeting, and then in 2002, the PMSF Foundation was formed, and they decided to name the syndrome after me and Heather McDermid, which was always a great honor. And I've continued to travel around. I've been over to see Thomas and Carlo and Chiara, and I haven't been to see Connie yet. I don't know why. She hasn't invited me, but I go all over the world, and I get to meet families and all over the United States and Canada. And it's been a great privilege to meet everybody and talk to them and, have a great time. And to talk to them about their concerns with their children or their family members, and to try to help them. Because a lot of the families, the doctors at home still don't know about the syndrome. They haven't heard about it, or they make an appointment for 6 months from now, or they have to drive 5 hours or something like that. Whereas they can call me and I can talk to them, or we would get on Zoom or whatever, and I talk to them. So you families out there, remember, you can get in touch with me with very little notice. Always available. So that's what I do, and that's what I still do. I'm interested in the clinical features. We're still, I've collaborated with many of the people on this screen over the years, and I'm still collaborating with a lot of you now, right, Thomas and Luigi, and Audrey. So we're still working together to characterize the features and to increase patient support. And it's why it's important. The families already know why it's important. You want to know what to look for, what the future holds for your children, and you want support for yourself and your other families. I don't have to tell you why it's important. That's fine. Thanks.
Thank you so much, Katie. And I know that you are a constant resource to families and to us. I would say at least once a week, I'm in contact with Katie with, family questions related to genetics or otherwise. And she's always there to help, and is frequently rewriting or updating a lot of resources online. So if you first found, the Rare Disease Nord website or something like that, it's very likely that Katie wrote that. So those are just additional things to everything she just mentioned. And she's probably being too modest because she's also on publications, all the time from this whole group. So thank you so much, Katie. Next, we have Connie from the Netherlands.
Thank you, Kate. I'm trying to share my screen. Hope this works. This one. Share. Okay, can you see my screen now?
Yes.
Okay, good. So my name is Connie van Avens. I am a clinical geneticist in Groningen, the Netherlands, and I coordinate the Center of Expertise, which is a European Union accredited center for Phelan-McDermid syndrome. So we fulfill the requirements that you have to have according to European Union to call yourself a Center of Expertise. We have in our center slightly over 100 individuals with Phelan-McDermid syndrome, both kids and adults, that we see regularly. That means every 1 to 3 years. And with a group of professionals, including psychiatrists, psychologists, pediatricians, neurologists, etc. And also I'm coordinating a European Phelan-McDermid syndrome consortium. It's working on a European guideline for Phelan-McDermid syndrome. And I will tell a little bit about that later. So I'll try to get on my next slide. Yes, there it is. Our journey started already a long time ago, 2016. We published our first paper, and then we had studied a group of 34 children that we have seen for slightly over almost 2 years, I think. And we studied their behavior and development, and the first paper was on their developmental aspects. And we showed in this paper already what's well known nowadays is that the children with the small deletions do better than this overall do better than the children with larger deletions. And there's a bit of a weird graph, but what you can see here is this is the cognition. So this is how well they develop, and this is their age in months. And you can see that the children with the smallest deletion do have a better development than the ones with a larger deletion. And we know that it has been replicated in other studies as well. So that is known. And now we have also recently published in the same group their behavioral aspects, and we place it into the context of other factors. So we don't look only at behavior, we also look at behavior in the context of communication difficulties, etc. We also studied the recurrence risk because we know several families with recurrence of Phelan-McDermid syndrome. So we wrote a paper on where you should, for geneticists, where you should be aware of when you have a child with Phelan-McDermid syndrome, how to assess whether there is a recurrence risk. So what you can see in this scheme, it explains which studies needs to be done after you have found the deletion in a child, the deletion 20q13. You have to do karyotyping to see whether it's a ring or a translocation, or it depends on which method the diagnosis was made. Sometimes you start with karyotyping, sometimes you start with microarray, and nowadays you usually start with microarray. And then we explained what studies should be done in the child, but also in the parents to assess recurrence risk. Then we did this clinical trial on the effect of intranasal insulin on the development and behavior in children with Phelan-McDermid syndrome. And that was because there was a very small German study performed, and families came to me with this paper, this German paper, saying, look, they have tried this insulin nose spray and they have good results, and we want this as well. And then I said, okay, they did it in a couple of children. There was no control. It was not placebo controlled, double blind. So if you want to do this, we have to repeat the study in a larger group of children, and we have to do it properly. So that when we did a clinical trial, and what we did, we studied all the children before we started with insulin for at least half a year to know what their natural development was. And then we looked what their development was under placebo or insulin nose spray. And that's what you see in this graph. So we estimated what the control growth rate was, growth rate in development, and also what the rate was under insulin. And so we could see what the additive effect of insulin was on their development. And we saw there was a slight increase, better increase in development under nose spray than when they used the placebo. However, there is variability. a lot of variability between children. So you need large groups of children to prove that this is significant. So we couldn't call this significant, although we saw that the effect was stronger in children above the age of 3. And in these children, there was a significant effect on cognition and especially on social skills. The problem was that after we did the study, a lot of families didn't want to stop with the intranasal insulin. So we wrote a guideline for this to do a trial treatment. So all children in the Netherlands with Phelan-McDermid syndrome and parents want to have the intranasal insulin, we can prescribe it under the condition that they have a test before and after half a year, so that we evaluate the effect of the insulin, and then the treatment is reimbursed by the insurance companies. So what are we doing now? We're doing a large follow-up study on the same 34 children we have seen before. We are now 8 years later, so I'm very curious to see how their behavior and their development has grown over the last 8 years. We have a study running on ring chromosome 22. There is an increased risk we know in children with ring chromosome 22 for a specific type of benign tumors. And we want to know how high exactly the risk is, because we have no information on that. We know there's increased risk, but we don't know how high it is. And we also don't know what the best way to screen for these tumors is. So that's what we're trying to figure out. We are continuing our clinical evaluation of the intranasal trial treatments, and we recently started a collaboration with Germany, with Ulm, because they also in Germany prescribe the insulin to their Phelan-McDermid children. And so we now want to combine efforts and to have the same, to use the same testing material and see with larger numbers if we can indeed prove there is an effect. In clinical practice, we both have the same experience, and that is that some children do not respond at all and others clearly respond, and especially they're improving communication. And last but not least, we're working on the European guideline for Phelan-McDermid syndrome. We recently had a consensus meeting where all recommendations from the guideline were voted, and everyone agreed with all the recommendations. And then these recommendations were shared also with the US group, because of course we don't want our recommendations to conflict with the ones that are in the US guideline. But of course we were, I think, quite on the same line, so there were no conflicts between our recommendations. The guideline will be published as a special issue of the European Journal of Medical Genetics, and the first parts of the guideline will become online available very soon, because we're already in the editing process of all these papers. We are working on a lay version that will become available in all European languages, so also in Spanish, for instance, which is also important for the United States, I think. And we are adding to the guideline a clinical checklist, so it's helpful for the surveillance of the children, and also an emergency card that can be given in case of emergency, so that you have a simple card saying what Phelan-McDermid syndrome is and what needs special attention in these children when, for instance, on an emergency ward. And what we also want to publish is the results of our international parental survey, and many of the families present here will also have participated in that survey. We had almost 600 participants, so it was great. And we will try to publish the results also as soon as possible. So thank you. That was what I had to tell in a nutshell. I'll stop sharing my screen.
Thank you so much, Connie. That was fantastic. Of course, we look forward to hearing any updates as soon as the clinical guidelines come online. Please send them to this group. Of course, we will circulate them. As you mentioned, they will be applicable no matter where folks live. So we're thrilled to be having a lot of literature on clinical guidelines coming out very soon. We appreciate all of your work on genetics, and of course also practical work of working with families to get them intranasal insulin. And that is just fantastic. And it also overlaps a lot with our emergency cards or neuropsychiatric cards. So we can also work together to give you any additional information if needed. So thank you so much. So next we have Thomas. I was so thrilled to see you recently. For all the folks on the call, I went to an international or European autism conference. And so I got to see Thomas in person and chat a bit, and that was fantastic.
Yeah, bonjour and hello to everybody. So I'm Thomas Bourgeon. I work in Paris, but I'm currently in Netherlands, in Nijmegen, Connie, for a PhD jury. So I'm closer to you. And so I don't have slides for the first time in my life. And but I will tell you a little bit about what we are doing. And first, I would like to congratulate the amazing team at the Phelan-McDermid Syndrome Foundation. I mean, it's so important. I mean, we found this SHANK3 mutation in 2007, and it was really, I think, important for the Phelan-McDermid syndrome. But I think the most important in that, if you have, I mean, what Katie has done with the deletion of 22q13 is that family can gather. And I think that's super important. I mean, that and to gather the family, you need a really great team of people. And I think that's the Phelan-McDermid syndrome is really an amazing example of what has to be done. Because I mean, genetics is great, but it's super important to have the people involved. So I work at the Institut Pasteur in Paris, and I'm basically a geneticist, but I would like to know what the genes that I study are doing in the brain, and I'm also looking at mouse models and brain imaging aspect. So as I said, I work mostly on SHANK3. This is as you know, it's a synaptic scaffolding protein. That is really a major gene for Phelan-McDermid syndrome. But on average, the patients, they lost more than one gene. And I think that's really important to keep in mind that probably there are some symptoms, clinical symptoms that are also associated with other genes and not only with SHANK3. So it's really important to get this in mind. And I think that's the main thing that I would like to solve in the very next year is how to modulate the clinical outcome and how to reduce the clinical symptoms. Because as you know, I mean, some patients they are less severely affected, some are more affected. We still don't know exactly why. Connie said that, and it's really a huge contribution that the size of the deletion of course matter in the clinical symptom. But for example, for epilepsy, we have no clear idea why there is about 60% lifelong children with and adults with Phelan-McDermid syndrome who will have epilepsy, and 40% who don't. And we still don't know. It's not really the size of the deletion. And so this is one part of my work, is to look outside the 22q13 region, outside in the genome, to see if there are other hits that might explain why some people are more affected and some people are less affected. So so again, I mean, working on genetics is, I think it's really great, but working on genetics without good phenotyping, without good clinicians and neurobiologists, I will say it's even dangerous to work on genetics without a good phenotype. And I think that's also what the Phelan-McDermid Syndrome Foundation is doing, and the registry that I had just had access recently. And this is really a great resource. And we are working together to see what, the resource, the time that the family spends to answer these questions. It's really important for the researcher, and we really acknowledge the time that the parents are spent to add this information. So because we need more information about this. So in terms of the things that we are doing now in Paris, so we are doing the whole genome sequence of many patients. We have more than 80 patients now with whole genome sequencing, but we also have some studies on iPS cells, derived neurons. You take some At that time, it was a little bit of skin of the patients to derive them in neurons. And we screened for 205 FDA-approved molecules, and we found that lithium was one of the compounds that could boost the SHANK3 expression. So you know that most of the patients, they lost one copy of SHANK3, but they still have one copy that is there. So if we can boost this one, it might help. And so we started after a lot of paperwork, and I have to say we need even more support from the family to get this paperwork time reduced, because this is really awful. And I think not only in France, I mean, in all countries. And but then we could start a clinical trial, a randomized clinical trial. It's a small one, 22 patients, and to test the efficacy of lithium in children with Phelan-McDermid syndrome. So it started on March. I mean, of course, we had COVID, but then we started in March 22. So there are already 6 patients enrolled, and hopefully in 2023-24 we should get the first results. And you can't imagine how impatient I am to see the results. Yeah, so we are working with Tobias Böckers at Ulm, and I think that's the field of the Phelan-McDermid syndrome is really people are collaborating and sharing the results, and I think it's really amazing. And in Europe, so I'm lucky to be in the dream team of CONI for the guideline of the patients, and also to make more effort even to know where the patients are in Europe, and then maybe to recontact them for future clinical trial. I think it's timely now. We have some ideas on what to do, and so we need to have the community working together. So I could talk 2 hours about SHANK3 and Phelan-McDermid syndrome, but I will stop now.
Thank you, Thomas. I think that's a great example, along with Connie just presenting, of really targeting all phases of research. And you're doing clinical trials, very late stage, clinical research, genetics research that's foundational as well. And we are so thrilled that any genetic information or some of the information we collect from families is useful for those studies. So a big thank you to all the families who go through that whole process, because it really does go to researchers. Thank you so much, Thomas. Next, we have Carlo.
Hi to everybody, and thank you for organizing this session, and especially to Kate. And it's a very pleasure to be here. And so I'm working in Milano, Italy, at the CNR National Research Council Italy, the Neuroscience Institute. And I started to work on SHANK since the gene was identified. So I was lucky to be in Morgan Schenk lab at the end of last cycle, so 1999, when the first papers about SHANK was published. So I contribute to show for the first time that SHANK is synaptic proteins. So I worked at the first paper, and then I contribute to show that SHANK is important for regulating the excitatory synapse. At that time, I was mostly working on another SHANK member, SHANK1. But since I came back to Italy in 2001, we start to work on try to characterize the function of SHANK3 using cell biology and molecular biology and neurobiology and so and so. And I was lucky that about I think 15 years ago, Chiara joined the institute, and together we worked. And now Chiara made a lot of contribution on the functional SHANK3. So I'm still expert on synapse. So what my major work is to study the assembly and the function of synapse. And as you know, there are many genes that are also deleted in neurodevelopmental disease. There are synaptic proteins. So my lab has studied some of these genes. But more recently, as Tobias, Thomas mentioned, there are other genes that are deleted in the 20q314 region. And so we, as Thomas, we think that this gene strongly contribute to the phenotype of the patient. And since up to a few years ago, there was very little known about these genes. Now we know a little bit more. And so one of the things that I'm trying to do is to characterize the function of some of these genes. So I think the approach that is important to do is study the single gene in the context of the function of the synapse or the neuron function, and then eventually put together with the function of all of them to somehow characterize better the disease at the molecular level, of course. And so one of the genes I'm studying recently is called TCF20, which is a gene which is implied. It's probably it's not deleted in all the PMS patients, but only in some, and it controls is a transcription factor controlling the expression of genes that are involved in neuron development, and also recently patient with single mutation of this gene has been identified with neurodevelopmental disease, but the function of these genes is still not completely understood. So using neurons in culture, using PMS, using iPS cells, derived neurons, using mice model, we are now characterizing the function of this gene and the gene that because it's a transcription factor, so control the expression of many other genes. So we're trying to identify which gene in particular are controlled by this TCF20. And so as I mentioned, my lab is strongly collaborating with Chiara, and also we have a very long and very established collaboration with Tobias Boker and Michael Schoen in Ulm. We go several times a year to Ulm. We share several projects, Shank and other stuff. So I think we really on this part, I think it's enjoyed to collaborate with other labs, and this is really important for making big progress in science. I think that's what I want to say today. Thank you very much.
Thank you so much. We really appreciate all of the foundational work you've done to understand SHANK3, what it does, and then to also push boundaries of what other genes are important, what might they be doing, what other cell types might be important beyond neurons. And it's just, it's sometimes difficult to explain the research that's not in clinical trials because it seems earlier, but it's foundational and it's important to open up a ton of new avenues for better understanding the syndrome and how to treat it. So thank you so much. Following up with that, we have Chiara.
Thank you, Kate. First of all, thank you so much for organizing this meeting, and as others say, for all the work that the association is doing, because we know, as Thomas said, that the most important part with patients, but also with scientists, because we need a lot of input for family, is what you do and the association also in Europe are doing. and are all the time inspiring. And also last week when we saw the meeting with the FDA was so inspiring, because hearing from the family what are the medical needs and what is important, because then we study synapses, we study the mechanism, but sometimes it's really important to have some question and say, these are symptoms or problems that the patient has, to try to focus a little bit our research. So, as Carlos say, I was extremely lucky because I joined his lab as a student when he came back from USA, from Morgan Chang lab. And at that time, there was described the first case of PMS. And so Carlo decided to study a little bit better the role of SHAN3 at synapses and what SHAN3 do in neurons. So I started working on this, and then during the year, I decided to focus my own research on, especially on SHAN3. And what the lab now is doing, we are using two different models, mice model, especially the SHAN3 knockout mice model that we share with Tobias Böckers, that is collaborating a lot with us, and iPS cells, because as also Alex and others say, mice are extremely important because allow us to study in the entire organism. So the role of the SHAN3 in the brain, and we can study behavior, we can test drug and the role of the drug in the behavior. Otherwise, mice are so far from humans, so it's important to have also other models. So we establish also iPS model, and we try to confirm some of the hypotheses that we have in mice also in iPS cells. And in particular, in this year, we discover that SHAN3 modulates the activity of a glutamate receptor that is called NGLU5, and we found that increasing the activity, there are some there are some molecules that are able to increase the activity of NGLU5, and we hope that in the future there will be also real drugs that can be used to increase the activity of this receptor. And we found that increasing this activity, but in mice, was sufficient to rescue or at least ameliorate some behavioral deficits. And now we are also testing this activity in human neurons, and it seems that also in neurons, it is able to rescue some molecular defects present in neurons. So because we believe that this receptor is extremely important, what we are now doing is better characterizing the molecular function of this receptor related to the presence or the absence of SHAN3. And also, as I told you, we showed that increasing its activity is important. We tested only one dose of the drug, so we are now treating the mice for a longer time, mimicking the situation of a patient. And also, we think that Phelan-McDermid syndrome is a neurodevelopmental disorder. So the majority of the study are done in adult mice. What we are now doing also with the treatment is try to work on younger mice. So to try to intervene during the neurodevelopment and also to understand exactly what happened during this short period that is extremely important for the brain. On the other hand, also speaking with patients, it's important to understand what happens when the children grow up and when they become also older. So another focus that we now have is study the role of SHAN3 during adolescence and also during aging, because we don't know what's happening in brain in absence of SHAN3 during the late aging of the brain. So this is a huge collaboration that we have with Tobias Böcker, and we are trying to understand what happened in mice, but also in other models during the aging. And finally, as you mentioned, brain is extremely complex, it's composed by different cell types. So it's important for knowledge and also for designing new therapeutic approach to understand the role of SHAM3 in these different cells. And so for this purpose, we are now using different approaches. So we are studying excitatory neurons that are the better characterized because a lot of labs around the world are working on this. But we are also working on inhibitory neurons that are less than excitatory neurons, but they are extremely important in modulating the activity of excitatory neurons. So what we are doing, what we did is knock down SHAM3 specifically in these inhibitory neurons to understand what happens when SHAM3 is not there. And it seems that some specific deficits that we see in mice are related with these neurons. So we are now trying to better clarify the role of SHAM3 in these neurons. And finally, a project that we are now starting is related with a complete different type of cells that are not neurons, but are glial cells. This is a European consortium that was built by us, by Tobias Boker and other groups in Europe. And the idea is we discovered that SHAM3 is not only a neuronal protein, but is present also in other cells, especially is highly expressed in glial cells. Glial cells are cells that are around neurons. They support and help neurons in their function. And now it seems that they are really important to regulate the activity of neurons. So in this project, what we want to study is what SHAM3 is doing in glial cells. Our preliminary data suggests for sure that if you knock down SHAM3 in glial cells, again, you have a defect in brain. So we are sure that SHAM3 is extremely important in these cells. In this year, we hope to understand really what it's doing. And also, we believe also that for trying to define new therapeutic approach, glial cells are also easier than neurons because can be modulated. They divide, they are cells that can be modulated in different way. So we hope to find some new target that can help to ameliorate the problems and the deficits present in the pathology. So more or less is what we are doing and the collaboration that we have in Europe at the moment. Thank you.
Thank you, Chiara. I'm very much looking forward to the data in glial cells, astrocytes, and inhibitory neurons, everything that is a new avenue that we are trending towards. And you also brought up a great point about looking at a lot of different ages, either in the models or also in humans. And that's definitely something that's happening across the scientific field, all the way from industry to, these models in labs. So folks seem to be really focused on different time points and different ages and making sure that you can intervene as early as possible while also checking if interventions also help later. And those are questions that we get oftentimes from families is there hope later? And is there a benefit to earlier? So we're answering some of those questions with this work. So just FYI, we are at 1:33 right now. So we have a little bit less than 30 minutes for about 6 people left. So I apologize, I took up a little bit of time in the beginning, but we will try to get through everyone. We may not have time for questions, but we have the questions recorded. So please go ahead and put them in the Q&A and we will address them if we don't get to them. So next we have Chris Winrow.
Thanks, Kate. I will try to be brief, but again, thanks for organizing this. This is phenomenal. And I think all the sessions have been really great to share, the science and patient perspectives and things all the way through. I'm an industry scientist. I'm a neuroscientist by training and have focused most recently in translational research, which means we look at what happens in animals and cells and sort of work to figure out if that moves into the clinic. And that's where I've been most active. I'll give it a little bit of the connection there as well. But my real connection to the PMS Foundation is through Jonathan, my son, who is he has ring chromosome 22 and was born in 2003 in San Diego. And a couple short years later, I got to meet Katie and a few of the other early members of the foundation at the Society for Neuroscience, which is running this week, actually. So this was, I don't know, whatever it was, 17 years ago, and really found an awesome opportunity to interact with this foundation. My background is, as I said, is as a geneticist and a neuroscientist. The fun fact, my PhD was at the University of Alberta. The lab that I worked in was 2 floors above Heather McDermid's laboratory. And that was about 5 years before Jonathan was born. So the world has kind of a wacky way of working. But from there, we moved to San Diego. I worked at Merck Pharmaceuticals for about 15 years, responsible for discovering new targets for neurodegenerative diseases and bringing those into clinic, and had the good fortune to work on something that ultimately became a drug for insomnia. From there, I joined a company in Massachusetts, in Boston, working again in the CNS space, and have most recently brought that into clinical development. That's been studied in Alzheimer's disease. We've got a clinical study that's going there, also in schizophrenia. And we've also recently completed a study in a rare genetic disease for mitochondrial disease. And so that's my day job. In addition to Jonathan and his care, he is surrounded by a great family. My wife, Dana, is a physical therapist, and she's actually been part of one of these sessions on lymphedema. That's her specialty, and I know has worked with a number of folks in the foundation there as well. And Jonathan's aunt, Denise, is on the board and is doing a ton of work there as well. So we are really thankful for all the work that you do as scientists, and again, really appreciate the opportunity to bring the families in on some of the work that's going on, and just show the openness and interaction that's there for folks to always reach out and be part of what we're doing. So again, my thanks to the other committee members, and again, thanks, Kate, for arranging this.
Thank you so much. I was hoping that you would mention Denise and Dana as well, because your impact touches our foundation in so many different ways. Just as another small example, Chris was one of the first people to respond with a letter of support to the CDC for our ICD code. So these supports, week to week, are amazing, that help us to do what we do and review anything that we are putting out to the community. So thank you so much. Next, we have Billy Bennett.
Hello, everybody. My name is Billy Bennett. My daughter, Cecilia, has PMS. And I actually made slides just expressly for the purpose of showing a picture of her. I know I'm really biased, but she's pretty photogenic. So this is Cecilia and her very good friend, Florence, who is an American Girl doll. And she went to the, last week, she went and did her annual visit with the natural history study at Mount Sinai. And so I'm also a pediatric gastroenterologist and a clinical researcher at Indiana University. When Cecilia was diagnosed, I emailed Katie Phelan, and she emailed me back within about 30 minutes. Because, I work in academics, so I figure, okay, I can just email random people and maybe they'll write back. She writes back in about 30 minutes and then gets me connected with the foundation. And it has been one of the more life-changing experiences that I've ever had. And I think Katie talks down her own, involvement, but she is sort of the glue that has kept all this together for 20 years. She is heavily engaged with the community, despite being an absolute expert and have the disorder named after her. So I think we owe a lot to her persistence and her engagement with patients. That being said, that's where my research has really shifted. So for the past 10 or 12 years, I've mostly focused on clinical informatics research and patient engagement research related to functional GI disorders, so problems like constipation and irritable bowel syndrome. As it turns out, these are huge problems in people that have neurodevelopmental disorders. So it's like I was handpicked to talk about poop with PMS families, which I do on about a weekly basis. And so we have two recent studies related to patient engagement. One was a study funded by PCORI that focuses on how do families interact with subspecialists. And we specifically targeted the initial genetics appointment as this point that a lot of families feel dissatisfied or feel like it could be better. And so we did a series of qualitative sessions. So I'm the scientific director of our patient engagement core at Indiana University. And so we focused on, what can we change about it? What are the sort of key problems? And the two that stood out the most were this idea of being transparent about what is known and unknown, like really addressing the degree of uncertainty. So I'll give you an example. So Connie mentioned earlier that the data about this association between deletion size and degree of cognitive impairment. Well, so Cecilia's deletion size is well over 7 megabases, so it's very large. And she has a very high level of functioning. She has an enormous amount of language, doesn't stop talking. She gives you a constant stream of consciousness update on what's going on in her head at any time. And she has a high degree of functioning. And so, one of the things that parents really wanted us to impart, and I agree with this from the parent side too, is that we don't know as much as we would like to think, especially about what is the level of functioning that one person is going to have versus another. So, really not putting kids in a box as far as what their developmental outcomes are going to be. And the other really big one, I think this is the biggest finding from this study, was that structured referral to a support community. So having PMSF be the first point of contact, and there's examples of this in other genetic neurodevelopmental disorders as well, that the expertise, the true expertise, which is in this on this panel and on the medical advisory committee, that's where the expertise is. And individual clinicians, even really well-trained subspecialists, probably will have seen 0 patients with Phelan-McDermid syndrome. So really, getting families engaged with the right community is essential. So the other study that we just did recently, so this was an R30, an NIH-funded conference grant, and part of it is that we did a survey ahead of the conference. So this is like a multi-stakeholder conference that included clinicians and scientists and industry, as well as families. And so we asked a bunch of different people. We got about 800 or so responses, and they ranged all across the board as far as these different syndromes. But PMS was the most common syndromic a form of ASD. And then we found, unsurprisingly, that this population has an enormous amount of GI problems. Constipation is probably the most dominant one. And what we hope to sort of bring out of that conference was this push to develop better clinical measures for individuals that have ASD and have neurodevelopmental disorders. We're trying to use something called the Rome criteria, which is how we diagnose a lot of GI problems, specifically functional GI disorders. And these are all validated in individuals who are neurotypical and who can talk, who can tell you that it hurts and where. We really need to focus a lot on better ways to measure clinical outcomes, especially in the GI system, because it's a heavily innervated organ. And so, of course, its effect, the effect of SHANK3 in your GI tract can be as profound as it is in your brain. And I think I'll stop there. And I just want to say that this is a truly impressive community of individuals. And I'm still shocked that I am even on this panel at all. And I just thank all of you, as a parent, thank all of you for how hard you work on something that is the most important thing in my life.
Thank you so much, Billy. I just want to highlight again how patient-focused the work is that Billy is doing, convening families about GI issues to have them even be able to tell us this survey doesn't capture my child. You know, we don't know if he's in pain or not. And, you know, capturing information about genetics, whether or not families are happy, frustrated, getting the right information they need early on. I was able to read through the whole report from that conference, and there was even activities such as love letters and breakup letters to genetic counselors, and it was just so educational. And so we're putting together a lot of those things now and writing them up in publications that will be shared with the community and with this committee. So thank you so much, Billy. And Billy has to put up with me probably once every 2 weeks in meetings just to talk these things through. So we appreciate your time. Okay, so next we have Michael from Germany.
Hello to everyone, especially to those who are associated to someone with PMS. I'm so happy to meet families again, and I hope for more exchange in the future. I maybe some of you I've met already because I've been to Orlando and New York to family meetings before. I also tried to share my screen. So first of all, my name is Michael Schön, so you can call me Michael. I'm a physician, and I'm a member of the SEC since 2021, so I'm the new one here. So I would first like to talk about where I'm from. So the city is called Ulm. So even Germans ask many times, where is Ulm located? So don't be ashamed if you don't know it. It's a very small but really cute city. We have the highest church tower there, and we also have a celebrity coming from Ulm, that's Albert Einstein. So I think it is, to introduce myself, I think it's best if I'm introducing what I would call the Ulm triumvirate. So I think it's kind of a unique model that we have there. It is one for all, it's the Institute for Anatomy and Cell Biology, headed by Professor Burkers. And he's been doing research on shanks, so their functions, the basic science on that, with different models such as mouse models and iPSC-derived neurons for more than 2 decades. And already during my medical studies, I came in touch with him. And since I've always had a big interest in medicine for children and also neuroscience, these two things came together because he was researching or doing research on Phelan-McDermid syndrome. But for me, it was also important when I started working that we come closer to the actually humans with PMS. So Tobias and me, we had the idea to establish a clinical expert center, which you see here. And this exists since 2015. It's built up by an interdisciplinary team and headed by Dr. Sarah Jesse here. And we have seen more than 100 patients with PMS so far. And the people from all over Germany, Austria, Switzerland come, also sometimes from other countries. We also initiated an association, mainly covering also families from these countries that I've mentioned. We also have some from Belgium and Netherlands. So Connie, we don't want to take them away from you. And I'm also a board member of that association. And also we have been visited by Katie and Sue, which was super nice some 3 years ago. And I hope that will also happen in the future, that some of you will also visit us here in Ulm or close to Ulm. So after so many years of basic research, we want to now extend this basic science towards more drug research, because what I think, and this is a general topic that came out here in several of the others introductions, was that that usually many people say that if you have a genetically induced intelligence deficit and autism, there's nothing you can do. But actually, I would say that is not true. And so the thing is, and maybe if I can explain this a little bit to you. So if you see, like I just draw a synapse. So this is where neurons communicate to each other, and the signal comes from here to there. And at this site of at the post synapse, so-called, there is the SHANK3 protein located. And but if you have a lack of SHANK3, such as in the condition of PMS, the signal is too weak, the synapses are too small, and the synapses themselves, they get rigid, so they're not so plastic anymore. So what can be done? Well, there are actually really some drugs that can maybe in the best way try to bring the balance back to such a situation, so that the synapses has a better signal and is much more plastic. And this is by different drugs that can either work very precisely, so that so directly act in increasing SHANK3. Thomas mentioned that before, like lithium. And there are also other drugs that are acting more broadly, like that they make the whole brain bring it to a more mature state. So we want to study these different drugs comparatively in our models, especially our mouse model, and see how they work, which strengths and weaknesses they really have. So, and I also want to finally point out to what I would call the essentials for the future now. And I think this has been also mentioned in many of the other introductions before. What I think is for the next, let's say, years to come, so important. So first of all, I think it is very important that we implement standards for diagnostics and treatments. And that's what Connie has been, or Connie's main topic has been about, that we implement guidelines. We have done that in Europe, and I was so happy that Connie also invited me to join this team. And and we also are in touch with the American team. So maybe we can once bring up a global guideline or so. That would be really here. And as you've seen, this is really, I mean, you have your family's collection and connections, and we also, like with Thomas, with Connie, and with Carlo and Chiara, we have a really good collaboration Europe-wide, and that's very important, I guess. And also the topic about, we want to know, we know very well at a certain stage how a PMS individual, what she or he has, but it's really important to know that over the whole course of life. And that's, I think, also very important. I think it's very important to also find biomarkers. It has also been mentioned before. This is also one of our main focuses we want to have in our institute with basic research, but also we want to have a look at that in our special clinical center. So, and also in cooperation with other centers. So also get in touch with me if you think we can collaborate on that. And so then I think in the future, and next year we are organizing a symposium in Europe, European symposium about therapeutical options in PMS. And then hopefully we come out with clinical trials. Especially, it would be best if we do that internationally, and thereby we will see which drugs are really helpful and for which phenotypes. And so it's really a privilege to be part of the SEC. And thank you, Kate, and to everyone.
Thank you so much. You've been such an addition in such a short amount of time, and you've brought so much to us. And we really appreciate the clinical work in Germany, but I also really want to highlight that diagram that you just put up there of the synapse where neurons communicate and how signals are exchanged, and how treatments might be addressing, something really specific there with SHANK3, or might be addressing a bigger picture. And I think that's something that I always try to communicate with any summaries that we do of anything. And I think that was really well done. Thank you so much. All right, we have 3 people left, and I'm so sorry that we're running over time most likely. So if you're one of these 3 people and you have to run, please message me separately. Otherwise, I will just call on you. But next we have Luigi, who just had a fantastic also conference session with their Clemson research initiative. I encourage everyone to go check that out. They're doing so much and have published so frequently in Phelan-McDermid syndrome recently.
Well, thank you very much for that flattering introduction, and especially for organizing all these. And thank you all, the people on the board for the great work. I do have a few slides from the same presentation that you mentioned, if you don't mind, but I promise I'll be very, very quick. I may share. So first of all, I'd like to share the credit, actually to delegate the credit, to give the credit to the other people that are part of what we created here, the Clemson University, the Phelan-McDermid Syndrome Research Initiative. You can see here the faces and the names of the people that are working on the syndrome, coming from different areas of expertise. as many of you have pointed, I just would like to do the same and show where we are. So we're in South Carolina, and we're at Clemson, that, as you can see, is very close to Greenwood, where Katie used to work at the Greenwood Genetics Center, and also Greenville, where there is a satellite office at the Greenwood Genetics Center where Curtis Rogers works. And actually, I may take the liberty to say that Katie and Curtis are honorary members of our Phelan-McDermid Syndrome Research Initiative, because we benefit so much from their guidance and expertise, and we really enjoy working with them. What do we do? These are some of the current projects that are either completed or ongoing. As you can see, we have two main areas which we're focusing our efforts. One is genotype-phenotype correlation. And I'm very happy to say that some of the first traits of the syndrome where we focused on, like seizures, sleep disturbances, and behavioral disorders, were selected by the families. This tracks back in time, and probably 2014, one of the conferences where we had a roundtable and families said, what are, if you had a magic wand and you could change something in PMS, what would you change? And these three ranked at the first three spots. So we're very happy to respond to the families' calls and try to meet their needs. So we're working on correlating these phenotypes with genotype when it comes to seizures, sleep disturbances, kidney disorders, behavior disorders, mostly autism, lymphedema, head size. Some of the families probably remember that we took some pictures with a 3D camera. We're finalizing that study as well. Speech and language impairment, neurological features, particularly working on some bioinformatic approaches. And then the second main area is a functional characterization of metabolic response. So we're looking now at particular response to IGF-1 and HGH, because these two compounds have been used in several clinical trials. And so we're interested in trying to identify subpopulations within our PMS cohort, depending on how the cells from these individuals will respond to these compounds, and eventually identifying the best candidates for clinical trials. In terms of what we recently published, there was this paper on seizures. I'll go very quick, I promise. We were able to provide a clinical characterization of the different types of seizures in PMS. You can see that some of them are more frequent than others, but basically you can expect any type of seizures reported in this population. And then by looking at signs or behaviors associated, again, you can see there is a vast array of behaviors or of traits that accompany the seizures episodes. In terms of genetic etiology, SHANK3, of course, is the main gene. We have a high prevalence of seizures in individuals with SHANK3 variants, but also in individuals with large deletions, particularly larger than 4 megabases. And this was quite interesting because it allowed us, you can see here with the black bars, the people with deletion sizes, the people with seizures, and the bars are the deletion sizes, and the gray bars are the deletions in people without seizures. And you can see that most people have deletions larger than these threshold. Most people with seizures, and most individuals without seizures have deletions smaller than these. And this was leading us to this other gene, TBC1D22A, that we believe is a good candidate gene for individuals with deletions larger than 4 megabases for the risk of developing seizures. We also looked at the metabolic profile. We noticed that there was a generalized reduced efficiency of major metabolic pathways, so it's something that affects the capacity of producing energy. And once again, there were more severe metabolic abnormalities in individuals with larger deletions. So in conclusions, we had some types that are more frequent, but basically any types of seizures may be expected in PMS. SHANK3 variants or even loss of this TBC1D22A gene may be associated with increased risk of developing seizures. There are disruptive metabolic pathways. Then another area of interest, sleep disturbances. This was a review of the scientific literature. We noticed that sleep disturbances are very common in PMS, considering all types of disturbances. Up to 90% of individuals, especially adults, experience at least one type of sleep disturbance. SHANK3, once again, is the main candidate. So we're talking about neurological traits. SHANK3 is definitely the main gene. And considering the impact on the quality of life and the high frequency of these disturbances, we suggest that individuals with PMS, all individuals with PMS should be assessed for sleep disturbances. Another thing, I think we are now starting to look at PMS as a multisystemic disorder, so involvement of many other systems in the body, not just as a neurodevelopmental disorder. And I take this opportunity to answer one of the questions there very briefly. Andy mentioned, what do we know about the SHANK3 unrelated PMS? And I think that depending, of course, on the areas that are involved, I think that as SHANK3 is the main gene, the principal gene leading the development of neurobehavioral problems, in the SHANK3 unrelated, we might expect more multisystemic problems, because we do see these kind of genes that are more proximal, and they might be related to problems, in this case, in kidney development and function. And there is another study that we have submitted for publication that was based on data from the PMS Syndrome Data Hub, and it basically validates the data from the literature. And we think that as a next step, it will be important to expand the genomic investigation and look beyond the 22q13 area, just to see other genes that may contribute to this phenotype. This is what I have. Hope I stayed in the time.
An unbelievable amount of work, and I think we can all appreciate that you are responding directly to major symptom areas that matter most to families, and that is amazing. And it's hard to even summarize, because there are so many ways that you contribute, including attending every conference we ever have, being a support in every way, PMS Awareness Day, everything. So thank you so much, Luigi. And we already got a question in the Q&A just about how to access these studies that I'll respond to directly and make sure they can see all your studies and also the summaries we've put out for them. But
I'm not as quick as Katie to respond to emails, but I'm doing my best. So
pretty close, if not. But I recognize that we are over time, so any panelists that need to drop off, we completely understand, and we have 2 people left, and I am so sorry that we are running over, but we're still very happy to hear from you. The last 2 people I've actually had the pleasure of seeing recently, and last but not least, so next we have Eva Law. She is showing up as William Bennett, but I think maybe that was just our fault with the links logging in. So sorry about that.
No problem. Thank you very much for having me. And of course, I completely understand if people have to log off. I also have a few slides. Great. How can I make this bigger? I should really know this by now. Okay, great. Yes. So my name is Eva Lord. I'm a cognitive neuroscience at the Institute of Psychiatry, Psychology and Neuroscience at King's College in London. I'm relatively new to Phelan-McDermid syndrome compared to several of the experts here on the panel. My main research area is autism research, and I've been involved since 2012 in a large-scale European initiative aimed at developing precision medicine for autism. And as part of this portfolio, we had a set of studies with autistic children and adults to identify biological markers or biomarkers. But also we had a preclinical program to identify new treatment targets using both animal models and cellular assays. We were particularly focusing on genes involved in synapse development and function, which includes SHANK3. And I went to a conference, and I was at a dinner in the evening, and I was sat next to Sue Lomas and Megan Boyle, and they had attended my talk. And they said, you know, how come that you are in a consortium that is looking at, you know, novel treatment targets and biomarkers, and you have preclinical work on SHANK3, and you've got clinical studies with autistic people, but you don't have a natural history cohort with people with Phelan-McDermid syndrome. And I came away from this meeting, you know, being so inspired by both of them, that I contacted Thomas. I don't know whether he's still on the talk call, and we wrote another additional grant straight away after this meeting to include a new much smaller cohort on Phelan-McDermid syndrome, and also get some more money for genetic whole genome sequencing. And so our study is called Synaptic Gene Study, or we call it Synergy, and it's embedded in this larger research program focused on identifying biomarkers in autism. And so this slide here is really an overview slide on this part of what is now aims to trials, which really goes from infancy through to adulthood using a lifespan approach. And what we have in common across these different linked studies is that each volunteer is characterized at multiple levels, from comprehensive clinical characterizations through to numerous biomarker methodologies, such as cognitive tests, brain structure and function, molecular markers, and genetics. So Synergy now is comparing two syndromes, Phelan-McDermid syndrome, and it's mainly led by us at KCL. And then also we have a sister study led by Louisa Gallagher in Dublin predominantly on orexin 1. And so what we're hoping to do is to establish or to contribute to a better understanding of the nature and frequency of autism among people with Phelan-McDermid syndrome, but also specific features, clinical features, and mental health and health comorbidities, and particularly to identify biomarkers that are related to synaptic deficits and try and trace how they are linked to behavioral and clinical characteristics. And then the additional goal is to see whether any of these biomarkers are shared or different between people with Phelan-McDermid syndrome as well as other kind of autism subgroups. But we also want to understand the variability or diversity among people with Phelan-McDermid syndrome. And now that we have just received through TOMA the whole genome sequencing from the first set of volunteers that we've seen over the last years, we are really interested in linking that with the genetic profile and look also at second and multiple hits. So I don't really have to tell people here that it's difficult to test or to include people with Phelan-McDermid syndrome in these sort of clinical research studies because of great variability in terms of age and also ability levels. Many children, of course, have profound special needs and needs which might make it difficult to include them in relatively routine assessments. So what we are doing is we have been capitalizing on the infrastructure that we have through AIMS trials in order to have a study assessment for each child or each volunteer that is tailored to their age and ability levels, using a kind of branching logic so that we're using tests that are most appropriate for each participant. The other thing I want to highlight here is that in terms of cognitive testing, and with cognition, I don't mean only, executive functions and attention, but also things like sensory processing, social processing, reward processing, emotional processing, but also things like understanding predictability or unpredictability. For many of these domains, we do not have well-validated tests and tests that are suitable for people with special needs, children in particular, with severe to profound intellectual disability are probably the most ones that are left out because we don't have adequate tests for them. So what we've been trying to do through a Simons Foundation funded project is to develop new touchscreen tasks to assess these different domains, to develop kind of, if you want, some biobehavioral profiles across these domains for each child, for each person, using mainly animated cartoons, and we're trying to vary them using artificial intelligence so that task difficulty levels and also surface features are again tailored to individual preferences and needs. So that's one project that is in process. Maybe just to mention that some of these subtests also kind of try to adopt a translational approach. So we try to back translate tests are used in animal model work so that we can directly compare that with work is done in animal, or not directly, but better compare it to work that's done in animal models. And the other thing that we're doing now is we've started doing MRI scanning with people with Phelan-McDermid syndrome. Again, most of you will know how challenging it is to scan people and children with special needs due to, the scanner is not particularly friendly, it's loud, it's claustrophobic. We've already tried to make it a bit more child-friendly through these spaceship tents that we're using, but we're also now working with MR physicists to implement a silent sequence that makes it hopefully a lot more calm and less threatening and uncomfortable for our children and volunteers. And the last bit I wanted to mention here is work by one of my postdocs, Jennifer Cook, and she's really interested in the parents of children with Phelan-McDermid syndrome, and she started a sort of like project that uses an online survey. It's a pilot project that we've just completed in the UK, looking at mental health and quality of life in parents of children with Phelan-McDermid syndrome, and looked at, and compared that between parents of children with Phelan-McDermid syndrome and autism, and looked at factors that might contribute to that, including the child support needs, autistic features, but also parental autism features. And our preliminary results suggest that diagnosis per se does not relate to parental mental health or quality of life. It's not necessarily that parents who have a child with Phelan-McDermid syndrome, all of them report lower quality of life. And interestingly, so far, it seems it's mainly the parental own autistic features and the strengths of autistic features that are related to mental health outcomes, but not quality of life, rather than the severity of other sort of support needs. And I probably stop here for now.
Thank you so much, Eva. There were so many themes to touch on there, but just a few of them. I think there are definitely trends towards comparing Phelan-McDermid syndrome to other disorders and syndromes, as well as autism in general, which has a ton of power. So we really, really appreciate that in the work, and also new assessments that can be touchscreen or be done at home, which is making it a lot easier for families to engage and always considering, what our population can and can't do, and heterogeneity in there, and even just decorating around the MRI machine is really nice to see. So thank you so much for that fantastic update. Last but not least, we have Sheldon, and I was thrilled to be able to meet Sheldon in person at the recent NORD conference, which is a conference for rare diseases.
So thank you, and thank you everybody who's able to stick on for a little bit longer on this. So I feel like unlike almost everybody else that's presented, I'm definitely more of a generalist than I am someone that's spent a long time working with Phelan-McDermid syndrome. I was introduced, well, let me take a step back. I'm a scientist at Rogers Behavioral Health. It's a very large behavioral health, mental health care system spanning 21 cities around the United States. And most of my work is building a biobank. So we have a genetics biobank that we're just launching that's tethered to all of our patient records. So we're not getting just a simple diagnosis, but we're able to look longitudinally at our patients that are receiving care and understand what's working and what's not. I was originally introduced to PMS through a NIH-funded project. I was working in a pharmaceutical company. I've heard a little bit about the glial cells and astrocytes. We had or have a product that really targeted those astrocytes, and for reasons unrelated to the science or people's interests, unfortunately that program paused, and there was the company took a different direction, and that's when I went into healthcare as a scientist. And while I've joined now my second healthcare system, I keep on coming back to something that I think is really important to Phelan-McDermid syndrome, which is the diagnostic odyssey that our patients face. People affected by these conditions can spend months, years, and even decades. In fact, in the state of Wisconsin, we have a regional group, and it's my understanding that the last 3 people that were diagnosed with Phelan-McDermid syndrome were older, 2 of them in their 20s, and 1 was 33 years old. And I have a massive problem that someone, and I don't blame anybody, but I have a massive problem that someone could go 33 years without having a diagnosis for a condition so impactful as Phelan-McDermid syndrome. And certainly I've worked with, as a generalist, I've worked with actually quite a few different rare conditions. And so in my, PMS has really been pulled into the fold for everything I've worked on. And in my previous healthcare system, we started to develop a product called Infogene, where we're looking at using genetic testing. PMS was on that, when I left it, on that panel. And so everybody born within the healthcare system would be screened for about 365 different conditions, of which PMS was there. That's a long road. I don't know when they would implement that and who's taking it over. But I've taken that same approach within mental health. And so we've identified over 50 conditions, and certainly PMS is one of them, where people will go these long extended periods of time without a diagnosis. And if they enter our system, and maybe it's the autism, maybe it's mood or behavioral issues, and with other rare conditions, OCD, repetitive behaviors, schizophrenia, depression, anxiety, all these things, you have patients entering the system, and they're pretty severe if they're coming to Rogers, and no one's really done any genetic testing. So we've really started to rethink how we're going to approach that. Many patients that enter our system are called treatment resistant or medication resistant, and they just haven't responded to the traditional therapies that maybe their psychiatrist or primary health care provider has attempted. And so we have a very heavy behavioral focus that's pretty successful. Okay, and I have literally over 100 years of success with that. But when medications are prescribed, I think it's really important that our patients are getting on the right medication. And even if one works, maybe there are 5 others that would work much better, especially if it was clear that they had Phelan-McDermid syndrome or one of the other 50 that we've identified so far. So really there's a heavy effort in trying to understand how to deploy and develop a genetic screener. So it wouldn't be a diagnostic, but it would be a genetic screener. I will admit, I'm a neuroscientist, I'm not a geneticist. I have no clue how to do any of that, but I know it needs to be done. And so that's what keeps me up at night. And that's why I ask a lot of questions from a lot of people that know a lot more about that than I do, to try to figure out how we could actually optimize that. And maybe my dream is every patient entering our system gets this genetic panel for these conditions. So, full circle, back to the biobank we're building, we're looking at a lot of collaborative opportunities for that. It's really meant to be a repository But one of my goals with some of that genetic material that we'll collect is to really look at the impact of these patients that are affected by, so many of these conditions on our system. And I'll leave you with this one final thing. And this is a publication we came out with in 2021. And we looked at rare and genetic diseases broadly. And we looked at healthcare utilization by people affected by rare conditions. What we found is nearly half of all of healthcare consumption was by people with rare conditions. And so that included inpatient readmission, emergency only. We weren't looking at outpatient or anything like that. We weren't even looking at medication costs, which we know orphan drugs are pretty expensive. But that is such a massive impact. And the subsequent papers that have come out elsewhere have shown a lot of that has to do with that diagnostic delay. So not only is it a quality of life for patients, but there's a really big hospital payer inpatient driver from the cost and utilization. So with that, thank you so much for sticking around, everybody.
Thank you so much, Sheldon. I think that speaks to an issue on families' hearts, all the time, which is the diagnostic odyssey and getting appropriate testing and getting appropriate support. So if any of that helps to identify more families, that helps us all. And, any of the public health work on some of the burdens of Phelan-McDermid syndrome also is just foundational and really important for increasing supports in every category. So thank you so much. And thank you to you all for staying. We had, a lot of the families that joined early have stayed this whole time, and our panelists have stayed this whole time. We've been on here for a little bit over 2 hours since 12:15. So I can't thank you enough. Every time that we have these meetings, I am reminded of how inspired I am by you all and how thankful we are for you all. And I don't know if families always realize that there are people all over the world working, every day on projects related to Phelan-McDermid syndrome on every part of the spectrum of research. I'll close with just 2 thank you comments in the chat and one question that we won't have time to address, but I will take it in and figure out the best way to get the answer back. But the first one is from Andrew Mitz, who says, thank you. After decades of encouraging scientists to work together on PMS, it means so much to me as a parent to see such great collaboration with each other and with the foundation. Other parents should know that we have exceptional scientists working on PMS. Thank you all. We have another comment from Abby Kraut, which says, I don't have a question. I just want to express the sincerest gratitude for the work every one of you do for our loved ones with Phelan-McDermid syndrome. The research being done gives us hope every single day. And another comment from Cynthia and from Maria. Thank you all so much for all you do. And no question, but just a huge thank you to all the panel for their work. So you are immediately touching families' lives, and a lot of the folks that will watch this recorded will feel inspired as well. We also have a question from Andrew Mitz, which is absolutely fantastic. I will find out the best way to answer it. But just so families know, if they can't see all the questions in the chat, it's a really great question, which says there's a lot of work being done right now on gene therapy for the SHANK3 gene. If successful, this means we will have a whole new generation of people with PMS SHANK3 unrelated, or people with deletions that don't include SHANK3. What do we know about this? How do we increase natural history study and all of these pieces of information so that families can know how to best participate in trials? This is a question that I'm sure this panel could talk about for maybe the next hour or so. And this is the right panel to ask because they all authored a beautiful publication that is inclusive and focused on folks with SHANK3 deletions and mutations, as well as, or variants, as well as those without. So we will get together and possibly draft a response and maybe have something around that, because that's a whole nother topic, I think, which is very important to address. So thank you to all of our attendees and our panelists, and for all the work that you do every day. Thank you so much.
Thank you to you, Kate. You're amazing.
Thank you.
Thank you.
Thank you.
Bye-bye to everybody.
Thank you. Goodbye. It was a pleasure.
Thank you.
Merci,
Bye.
Kate. Merci.
Bye.